Welcome to the Wild Side. This is your host, Kight, and that was Bonnie Bear with Creature Fear. And on today's show, I'm going to catch you up to speed with some of the most recent science news. And the first story I want to talk about is some research into how the gut feeling shapes fear. And what do I mean by the gut feeling? Well, if you've ever been frightened or nervous about something, you know that there are certain physiological responses that you tend to have. So for example, you might have a faster heartbeat, you might have sweaty palms, you might feel kind of a nervous or a knotted feeling in your stomach, and these all are things that we kind of refer to cumulatively as the fight or flight response. Well, the last one of these, that strange feeling in your stomach, is one that researchers have been really interested in for a while because they know that the brain is sending signals to the stomach, but they also know on top of that, that the stomach is sending signals back to the brain. And so there were some researchers who were interested in figuring out why it is that this happens and what the purpose of it is. So does this actually function in helping us to respond to these situations and maybe even to learn about these situations so that we know how to respond in the future? So one of the things that they did was focus in on the vagus nerve. So that is the nerve that is linking the brain and our internal organs in general. So the efferent nerve pattern is the one that goes from the brain to these organs, and then the afferent nerve pathway is one that goes from the organs back to the brain. And they wanted to look at both of these things. And by studying this link, the researchers were able to figure out how it is exactly that that gut feeling is impacting our fear. So first of all, what they did was they cut the afferent nerve fibers, so the ones that are going from the stomach back out to the brain, because they want to figure out what happens when suddenly this connection no longer exists. And they wanted to compare that to situations in which the connection is intact. And so if there's a difference between those two, then you can see what it is that this stomach signal, for lack of a better word, is able to achieve whenever it is triggering something in the brain. So they did this in rats, and then they placed the rats into situations that would normally make them nervous. So they exposed them to things like open spaces and bright lights and other things that rats just inherently and intuitively know that they should avoid because they are dangerous to them. So rats that had the snipped afferent nerves were much less wary relative to rats that had intact nerves. So this suggests that there are these innate responses to fear that are suddenly influenced by the difference in signal moving from the stomach outward to the brain. So this suggests that in some way the stomach is helping to tell the brain that it needs to be worried about these things that the rats are born knowing that they should be frightened of and should avoid. However, the rats were not completely fearless, so they were still able to learn appropriate fear responses over time, which suggests again that the afferent pathway is one that has to do with this innate stuff, but isn't as heavily involved in the learning stuff associated with fear. So animals, both with and without the afferent pathway, were able to eventually learn to fear this stimulus that they were presented with, in this case, a loud and unpleasant noise. Now, the noise itself was actually just, that's a negative thing because it was kind of frightening and unpleasant. But when they shifted that from a negative to just a neutral kind of experience, so they were put in this situation and then given another stimulus that shouldn't really make them, you know, extremely frightened or extremely unfrightened, which is kind of in the middle, they again saw a difference between the two types of rats. So those that had no gut instinct took longer to learn that the noise was no longer indicating that something bad was about to happen. So they'd all been through that experience in the past where the noise was a bad thing, and then suddenly they're put into a new situation where the noise doesn't herald a bad thing. So usually what they do in these kinds of experiments, and they didn't really describe this too much in the summary that I read, but usually what they tend to do is put test animals in an area and kind of give them a little bit of an electric shock because that's kind of a stimulus that is immediate. Sometimes they might spray them with water, or if it's a longer term thing, they might put them in that situation and then deprive them of food or something like that. So whatever the case might be, they did something so that this noise was associated with a negative experience, and that's when they saw these first set of responses. And then they did it again, and they made it so that when they played the noise, the bad thing was no longer happening. So again, the rats that didn't have the gut instinct took longer to learn that there was this shift now, so that the noise was no longer bad. It was actually just, it was either, all right or neutral, depending on how worried they'd been in the previous situation. Now, all of this fits with recent research that indicates that stimulation of the vagus nerve is associated with relearning. So not only does it seem to have some impact on this innate response to kind of fear-inducing situations, but also it has to do with relearning a new set of stimuli, and figuring out what it is that they mean and how it is that they're impacting your life and how you should respond to them. So what is the mechanism behind all of this? Well, the researchers think that the abdominal signals affect the balance of neurotransmitters in the brain. So once the signal is sent from the internal organs, from the stomach, back up to the brain, this triggers a release or maybe an inhibition of neurotransmitters that are in the brain. But this hasn't been studied yet in very much detail, and so they need to kind of explore this more in the future. They need to know exactly which chemicals are altered, how many are altered, what's the time span over which they change, and how does all of that then translate into complex changes in behavior, because the neurotransmitters are really just the beginning of a whole new set of responses, which is not just having that signal sent from the stomach up to the brain, but then turning that back around and making the brain then tell the body, whether it's the muscles or the nerves or the hormones. I mean, it could be all sorts of different things that the brain is then telling the body to do in order to respond appropriately and help that animal basically do whatever behavior is going to help it survive and be safest in the end, because that's kind of how all of this works. That is the evolutionary purpose of this sort of response, is to keep you safe and healthy for as long as possible. So what's all the relevance of this work, other than just kind of understanding how this process works? Well, it could be really useful in the field of psychiatry, and also in the treatment of people who are dealing with conditions associated with fear and stress. So it's very likely that these things that we're seeing in the rats are not dissimilar to patterns that happen within humans as well. So for example, if you've got people that have post-traumatic stress disorder, they might be able to undergo some sort of a treatment where their vagus nerve is stimulated, and this helps them to learn that certain triggers, maybe sounds that sound like a helicopter or a bomb going off or something like that, that they're no longer associated with negative outcomes, that these are just neutral things. So they might have been negative out when they were deployed somewhere or when they had this negative experience, but now in a new situation that's no longer a bad thing, and they need to retrain themselves, and potentially simulating this nerve could help them to do that. And this treatment actually already is in use for epilepsy and even depression, but I'm not sure that researchers really understand exactly how and why it works. So better understanding the effects of stimulating the vagus nerve and kind of how and why it works in the way that it does would potentially help people to use this treatment not only in new situations, but also to use it better in the situations where it's already employed. So moving on very much from kind of neurobiology and behavioral biology to something that's completely different. So I'm thinking about now genetics and whales. So I talked about whales fairly recently, and there's a brand new result about kind of some more research on whale subspecies. And this is something that I didn't really address much in the show that I did a few weeks ago on whales, so it's kind of a nice new direction to take on that topic. So we know that humpback whales are kind of one of the big, well, literally big, but also figuratively big species that people are interested in conserving. They get a lot of press, and people are quite aware that whales aren't necessarily doing all that well. And one of the things that a lot of researchers have been hoping to do is to better understand the genetics of whale populations. So if you did tune in a few weeks ago, you'll remember that I talked about how a lot of whales tend to roam incredibly widely. So they migrate between breeding and feeding grounds between the winter and the summer. And there's not really known how much the different populations of whales are kind of split into little pockets, how much they are actually all one giant thing that kind of intermingles, how far all the different individuals go, how much variation there is in that. And it's really important to get a handle on the different individuals and the different populations, because if you understand this, then you get an idea about gene flow. And gene flow is really important because we want to make sure that we've got enough genetic variation being preserved in the wild, and we also want to make sure that we're protecting all the different subspecies, because you might be protecting one group of animals or a group of animals in one location, but then actually neglecting a whole bunch of others that need completely different protections because they are completely genetically distinct, they don't breed with the others, they don't go to the same places. So you need to understand those kind of intermingling effects of genes and breeding and migration. And that is exactly what this new study was beginning to do. So researchers have used little biopsy darts that they deploy. So they find the whales when they're kind of up breathing near the surface, and they shoot them with the darts, and the darts chunk off a little bit of skin. The whales probably barely even feel it, considering how thick their hides are. And then the darts plop back out into the ocean so they can be collected, and then the genetic samples can be analyzed. So the researchers did this for humpback whales in the North Pacific, the North Atlantic, and also the Southern Hemisphere. So the three main places that these animals are found. And the darts collected both mitochondrial DNA, which is inherited only from the mother, and also nuclear DNA, which is inherited from both parents. And this is interesting because the two different things can provide different types and levels of information. So mitochondrial DNA provides insights into the movement, particularly of female humpbacks, from one population to another, and in particular over the last 1,000,000 years or so. And again, because mitochondria are inherited only from the mother, that's why this is very kind of female-centric. And this is quite interesting because we know that a lot of times, we know already that whale populations tend to be kind of dominated by these females, and the females will group up and they'll raise their calves together, whereas the males kind of go off on their own once they reach sexual reproduction age, or kind of as they're approaching that. So we know that the females kind of dominate this society in a sense, kind of like what we see in elephants. But we also know that in a lot of different animal populations, you do tend to have one sex or the other that disperses or chooses to stay put in order to then breed. And that allows you to make sure that if both males and females are dispersing, then you might find that they kind of run into each other, and then you have siblings accidentally mating. If they both stay, you might have the same thing. So there tends to be only one or the other that moves or doesn't move. So focusing on one particular gender allows you to kind of get an idea of which animals are staying put and which are dispersing out into the environment to spread their genes and reproduce. Now, the nuclear DNA provides information on the species as a whole because it's shared by all of the animals. It's not just from the females, but it's, of course, when you're breeding, you have some of the males and some of the females' DNA, and that's what makes the offspring. So that's why nuclear DNA provides more general information. So the results of the analyses, they were just looking at the similarity of the genetic samples taken in these three different broad environments. And what they found was that the females seem to have moved great distances over time from one hemisphere to another. And I'm not going to go into detail about how they're able to look at the timeline from the genetics, but basically it has to do with the fact that we know that certain types of certain segments of the DNA will stay basically the same over time and change very little, and other segments will change much faster. And you can kind of get an idea of the relative speeds of those two different things, and you kind of get a nice baseline reading so that anything that's different from that baseline, you can get an idea of, how different is it, and then using the speed that you've been able to estimate, you can then get an idea of how long did it take for that difference to emerge. And so that allows you to kind of count backwards and figure out how long ago something was introduced or taken away from a population, genetically speaking. So that's how they were able to figure out that it was females in particular that have been moving over time for quite a long time. In fact, they might have been moving from one hemisphere to another, so they're crossing vast distances. But even though they were doing that, the whales tended to stay in the same ocean in which they were born. So they might go from one hemisphere to the other, but it might just be from the South Pacific to the North Pacific. They're not going from the Pacific all the way around to the Atlantic. So basically what happens in this scenario is that it creates little isolated pockets of whales in each of the three study sites, which means that these three distinct populations aren't really intermingling that much and breeding with each other. They're not sharing their genes. They're quite obviously distinct. And the researchers, in fact, refer to them as being semi-independent. So every now and then you might have a whale that kind of goes from one of these to the other and intermingles and breeds, but for the most part, they are fairly distinct. And they seem to be showing completely different evolutionary trajectories, which again has to do with the fact that they aren't sharing genes, and also because the environments in each of these places, they're quite different. And so of course the whales are going to respond to that and be adapted to their local environments. And so between this kind of lack of gene flow and also specializing to the environment in which they live, you do have these distinct trajectories that are emerging. And the researchers were actually surprised by this because we know that these animals migrate huge distances between their breeding and feeding grounds. And so you would have thought that over all this space and time, all these guys would have been crossing paths and intermingling and breeding with each other. But maybe they don't. Maybe they use completely different routes. Or maybe when they do see each other, they just don't interact all that much. Maybe they don't even see each other as being relatives. And we know that this does happen even in species that look and act very similar, that there is enough of a difference that they notice it, and so they choose to just ignore each other. But at the same time, the researchers also noted that they were already aware that there were some differences, some pretty obvious differences between the populations. So for example, northern whales tend to have much darker bodies, and the bottoms of their flukes are much darker, and so there were some signs that potentially there was some differentiation. But sometimes those little physical things like that may not actually have that much to do with a lot of genetic difference. Maybe it could be all environmental, and so they weren't totally sure until just now that these physiological things and these morphological things also seem to be relevant in terms of the genes underlying them. So as I said, it's important to understand the connectedness of the populations in order to determine appropriate conservation measures, and also to think about the implications of environmental changes, because if these three different populations of whales are different, then as the climate changes, as anthropogenic disturbance changes, as all sorts of things change, then they might be impacted differently, and so that means you might need to have different management and conservation plans. So just to be even more specific, although we might see improvements in the numbers of one group, so for example the North Pacific humpbacks, that doesn't mean that all the humpbacks are experiencing a population boon, and that means that we might need to keep tight restrictions on certain groups that we otherwise might have said, oh, actually it's okay to start hunting these again, or it's okay to be a bit more disturbing in these environments. You can go out and do more whale tours because these guys have rebounded and they're nice and healthy. So it just allows you to know that you need to tailor your conservation and management schemes a bit more or a bit less. However, it is also important to note that the populations might shift in the future. So as I mentioned, climate change could come into play, and we know that whales will follow food, and that food might become scarce or might be relocated as the temperatures and the water conditions change. So maybe this could lead to different levels of interactions between the animals. Maybe this could even kind of drive them all towards one single place where they all find their food, and this means that they might suddenly start breeding, or it might even drive them further apart so you have even less interaction between them. It's hard to say until we actually can start tracking these things in the wild. But basically, in addition to fostering whale conservation, it's important to think about how these genetic results can also allow us to understand a little bit more about historical ocean conditions. So as I said, you can kind of work backwards using this genetic stuff and think about the time line over which these events happen, these female migrations. And when you think about the female, I should say actually dispersals rather than migrations, and when you think about the female dispersals, you can think about why is it that they're moving, and where is it that they're moving from and that they're moving to. And again, you can start to tease all this out using the genes, and also if you go back and start looking for maybe any fossil evidence that might be available to back that up. And this allows you to kind of think about what the different habitats were like at different points, and what the larger environment that those habitats were kind of set within, what was that like as well. And this, again, when you can kind of reconstruct the past, it allows you to make certain projections about the future. So, just like we take ice cores in order to think about certain effects of the levels of carbon in the atmosphere, for example, you can kind of do similar things in the oceans and think about what was it like in the past, and as we fluctuate, and as we might return to some of those levels because of various reasons, what will it be like again in the future? Can we kind of make some predictions? So this is useful not just for understanding some interesting things about whales and whale history, but also for thinking about the oceans and the planet in general. Welcome back to The Wild Side. This is your host, Caitlin Kight, and that was Robinella and the CC Stringband with Honey, Honey Bee. And on today's show, I am talking about some of the most recent science news. And the next story that I want to talk about is, predictably, about honeybees. So I want to think a little bit about what bees can tell us about the environment. Other than that, there are flowers nearby. So actually it is kind of about whether or not there are flowers nearby, but it's a bit more detailed than that and a bit more useful. Now, just to give you some background about honeybees and exactly how this particular sort of environmental monitoring might take place, I need to think about the honeybee waggle dance, which I may have talked about briefly before on the show, but I don't think in much detail. So scientists discovered a long time ago that the honeybee waggle dance is a behavior that the honeybees use in order to share information with their hive mates about where food can be found out in the field. So we know that when honeybees return from going out and looking around and finding nectar and pollen, that they use two different parts of their dance to provide information. First, they use direction, so they kind of orient themselves in a certain angle, and that tells their fellow bees where to go once they leave the nest. Are they going 30 degrees, are they going 60 degrees, etc. And then they also use the duration of the dance to indicate how far away. So the longer they're dancing, the farther their hive mates are going to have to go in order to find the food. And using a protractor and a timer, researchers can collect this source of information extremely reliably and extremely easily, and actually then go out and locate exactly where it is that the bees have been foraging. So this is a really amazing thing that scientists discovered several decades ago, and they have been using it ever since to think about how bees are utilizing the environment and how much they utilize certain types of landscapes rather than others, and so on and so forth. Well, now scientists have discovered that actually we can use that waggle dance to find out something else as well. And specifically, what they want to know about is whether the bees are able to use certain types of habitats very easily around them, and whether those habitats are kind of healthful and useful for the bees, and whether that is an indication of the overall health of that landscape. So what they did was, at 3 different hives over a 2-year period, they looked at a total of 5,500 dances. And consistently, the dances were indicating which type of foraging habitat was being most utilized by the bees. And this was done in kind of a mixed area. So there were agricultural areas, there were kind of more developed, kind of villagey type places, there were reserves nearby, there was a big mix of stuff in the landscape. And what they found over this entire study period was that over and over again, the bees were going to the same places, and that indicated that these were the best foraging habitats. And all of these little pockets of flowers were located predominantly within a national nature reserve that was set within a broader kind of urban rural landscape. So as I said, there were lots of options, but the best one within that whole landscape seemed to be the nature reserve. And specifically, when you're kind of looking at, when you kind of, graded the different types of habitat, I guess is the right word, as high level or organic level or whatever type of agricultural activity, what they found, surprisingly, was that it was actually the high level agri-environment schemes that were the best, and it was the organic entry level schemes that were the worst. So areas that had more actual intensive agricultural areas within them, in addition to all this other stuff, the nature reserves and the other types of things, whenever there was a higher proportion of this high-level agricultural stuff, the bees tended to forage better and find more stuff for them to eat. However, the least frequented habitats were the ones that were organic, and we might have thought that organic would have been best, but the researchers think that maybe it has to do with differences in mowing techniques, since the organic areas tended to rely on mowing rather than pesticides in order to get rid of the weeds. And actually the mowing was getting rid of the flowers, so the weeds, if you like, that the honeybees were particularly interested in. And so in order to find the blossoms that they were interested in, they instead had to go over to the more intense agricultural fields where they had the pesticides being used and the mowing was not being used as much, so they still had those things that they were interested in. So the fact that the researchers could learn this information from watching the bees suggests that actually you don't need to sink lots of time and money into large-scale, long-term habitat surveys in order to do quality and quantity monitoring of production. So if you have a landscape and you want to get an idea of how healthy it is, how much is available for organisms like bees in order to get their pollen and their nectar, if you wanted to get an idea of whether there is a lot, if they're having to forage really far, if one type of habitat is a bit better than another, all you could actually do is just look at the bees and see what the bees are doing. You could get an idea of exactly how far they're going and in what direction, and then go out to that site and see what it's like, and have an idea of which areas are healthiest, which are providing the insects with the most, and also that can kind of allow you to project to the future, because you know that where the bees are pollinating, then you're going to have more crops in the future. Now, might be a little bit different if you're thinking about agricultural stuff versus kind of wild things, but still we know that the bees are providing some benefits of pollination and even the really disturbed agricultural areas. So you can also project into the future to some extent or another whether that is going to be kind of a self-sustaining habitat that's going to be frequented and maintained by bees into the next several years. So this is quite a nice thing, because across the EU a lot of money has been pumped into improving the rural landscape. So something like 40 billion euros over the last 2 decades. People have been looking into ways to have the right mix of reserves and villages and agricultural schemes, and how heavily should we manage this plot versus that plot, and what percentage and what type of thing should be next to another. And so it all becomes very complex to get exactly the right balance to allow humans to get what they want out of it and to allow insects to get what they want. So now we know that the best thing is a reserve, but the next best thing after that is to have kind of a mix where you've got a reserve and also maybe some high intensity agricultural areas, which actually is really good news, because that means that the thing that is useful to the bees, the thing that's useful to us is also useful to the bees, and so maybe we're not sacrificing the insect wildlife just for our own gain. So it maybe means that we could kind of share space happily together and not be so selfish. But in any case, it does allow you to kind of start to assess these things and make some models about these things using the bees rather than using a fleet of people out there surveying the habitat on foot, taking all their time and energy and money to do that. And this is particularly interesting because a lot of these schemes, people have tried out lots of things, but then not really taken the time to collect data to see whether or not they've worked, or to compare the different types of areas and see which one works best. So this is a perfect time to use this new honeybee technology, if you like, to go out and collect this information and to kind of assess how things are going and what we need to do into the future. So now I'm going to shift gears again. So today I am all over the place in terms of topics, and the next thing that I want to talk about is the discovery of the oldest known sperm. And this is a story that has definitely made the news over the last week or so, so you might already have heard about this. But if you haven't, it's a pretty good story, so it's an interesting one to listen to. And this discovery has been made by a team of Australian researchers who were looking at the Riversley World Heritage Fossil Site in northwestern Queensland. And this is an area that approximately 17 million years ago was covered in rainforest, so it looks incredibly different than it does today. And they collected samples from a cave that at the time would have been filled with a pool that was inhabited by a variety of freshwater organisms, including shrimp-like crustaceans that are known as ostracods. And these are the crustaceans whose sperm was discovered. Now the cave was also inhabited by bats, and the bats, of course, when they're hanging from the ceiling, they release quite a lot of guano, And all of that would have enriched the pool and made it a really productive place for the ostracods to live, because the guano does contain a lot of nutrients that then could have been broken down by the ostracods and they could have been feeding off of it. Now, the side effect of the guano, other than just producing a nice meal for these little crustaceans, is that it also would have put really high levels of phosphorus into the pool. And this would have helped facilitate mineralization of the soft tissues that, in fact, were discovered and analyzed here. And if you know much about paleontology, you know that soft tissues are often quite hard to come by because they do degrade much faster and much more easily. And it's the hard stuff that we tend to find evidence of. So it's not all that common that we see things like fur or feathers or skin or organs or things like that. So this is one of the reasons why this is quite an exciting thing, is that they are able to look at these soft tissues and find a type of fossil that ordinarily would just be really difficult to locate because it would have vanished long ago. Another thing that's kind of amazing about this study is that the samples were actually collected in 1988, and it appears that they have not been analyzed until just recently. And this probably was actually quite beneficial because over the past 20, 30 years, technology has really advanced quite a lot, and so they're able to see things in much better detail now than they ever could then. And so that really allowed them to get good glimpses of these samples. In particular, they were looking at the internal organs of the ostracods, and this included sexual organs, which contained intact sperm, with the sperm nuclei, which of course would have once contained the DNA, and also something called a zinker organ, which is a chitinous muscular pump that is used to transfer the sperm into the female shrimp. So one of the things that was notable about the sperm, besides the fact that it was found at all, and also besides the fact that it's so incredibly old, is that they're very long. So they're about 1.3 millimeters long, which is something that you actually could visualize with your naked eye, which is not really what we tend to think of when we think of sperm. And what's even more amazing is that actually this is about the same length or a bit longer than the ostracods themselves. So these things would have been incredibly tightly coiled up within the sexual organs within the ostracods. And so it's quite an impressive bit of reproductive reproductive equipment, I guess you could say. And actually you can kind of make some inferences about the life history of these animals by knowing this, because we do tend to know that where there is quite a lot of competition between different males for a female, then the sperm can be quite sizable and quite powerful. So it may be the case that these females would have mated with multiple males, and then the sperm kind of battled it out within the female, and it was the males that had the longest and the biggest sperm that were able to actually end up having a successful reproduction with the female. So what's kind of cool about this site is that there are lots of other things that have been found in this same place, and this suggests that actually in the future there could be even more discoveries like this one. So for example, they've had soft tissues of things like insects and also marsupials. They've seen internal muscles, they've seen eyeballs, all sorts of unusual stuff that we don't tend to find. And there's also a similar site in France that, like this place, has had quite a large amount of bat guano that seems to have helped with the fossilizing process. And so the researchers think that actually this kind of gives us a pattern that we can then use in order to locate more samples. So we know that these caves that have lots of bats will tend to have samples of these soft materials. So maybe we can then search for more caves like this in order to find even better, more remarkable, more interesting fossils in the future. Welcome back to The Wild Side. This is your host, Caitlin Kight, and that was Lenny Kravitz with Fly Away. And just before the break, I was talking about some fossils, some interesting fossils that were found in Australia, and now I want to kind of stay in that region of the world and think about the geographical origin of the kiwi. Now previously, scientists thought that the kiwi's closest relative was the emu, which would suggest that the kiwi originated in Australia because that is also where emus live. And so it made sense that somehow the emu would get from Australia to New Zealand and evolve in some way that made it much smaller and nocturnal, and there you go. But a new genetic study suggests that actually kiwis are actually more closely related to an extinct bird from Madagascar. And this contradicts previous findings that suggest that kiwis were most closely related not just to emus but also cassowaries. So it's basically completely cut ties between the emu and all of these large rat-type birds that are living in Australia. Now the rat-types are this group of birds that are flightless, so things like the rhea, the elephant bird, the moa, the ostrich, all those kind of unusual dinosaur-looking birds that you've probably seen in zoos, if not in the wild. Now this was quite a contentious issue for New Zealanders because the kiwi is their national bird, and they didn't like the idea that it was an immigrant from Australia, or that it had, somehow these ties from Australia rather than being born and bred in New Zealand. So they're quite excited about these new results. And to go into those in more detail, the actual closest relative, this Madagascan bird, is one called the elephant bird, which would have stood 2 to 3 meters tall and weighed something like 275 kilograms. So obviously there is quite a big difference, not only between the kea and the emu, so that was already kind of a stretch, but even more so between the kea and its actual closest relative, the elephant bird. And researchers have been trying to figure out for years and years, actually for about 150 years, exactly what the genealogy of these species is. So as I said, all these guys are within a group called the rat-types, so things like the moa, which is an extinct bird from Australia, the ostrich, which we know is in Africa, there are a couple different species of that. The rhea, again, there are a few species of rhea, those are in South America. So these things are all over the world. They're on these very distinct continents that are currently quite far apart, and yet they all have kind of similar morphologies. Now the kiwi obviously looks a bit different from these other ones, but certainly the rhea, the emu, the ostrich, they all look very similar, and they all have similar lifestyles. They aren't flying, they live on the ground, they have kind of shaggy feathers, lots of similarities. It was previously thought that there was originally a single parent species that just maybe was on, Pangea, and as Pangea spread apart, then that parent species would have had little pockets of itself on each of the different continents, and those would have then evolved separately into the species that we see today. So that was kind of the idea, where you've got one parent and then lots of little daughter species as the southern continents drifted away from each other. However, this is refuted by the recent evidence, which indicates that the close relationship between kiwis and elephant birds happened despite their geographical separation. So specifically, the authors of the study suggest, they believe, that the rat-types must have dispersed around the world via flight, because New Zealand and Madagascar have never been all that close. They've only ever been distantly joined by Antarctica and Australia. So the only feasible way, really, for these birds to get from one to the other, or for an ancestor of these birds to get from one to the other, is for them to actually fly, because it would be highly unlikely that you would have at least one bird. I mean, you'd have to have a couple in order to have breeding and then descendants to get from one of these places to the other, maybe by floating on some wreckage or being blown off course. It's such a great distance, and it would have to happen at least a couple of times to allow the descendants to happen, unless it happened to be a pregnant female, I suppose. It's very unlikely. So what's much more likely is that you had something that was just simply able to migrate, to disperse, to fly from one continent to the other. And the DNA evidence suggests that this dispersal event, this supposed dispersal event, occurred about 50 million years ago, just after the dinosaurs went extinct, and just before the mammals took over and became the dominant terrestrial animals. So in between these two periods, you've got kind of a lull, during which the rat-types would have been able to exploit this opportunity. So dinosaurs weren't there to eat them, mammals weren't there to outcompete them, and so they had this chance to be kind of the large resource-utilizing organisms of the day. And so that's why you have these big ostriches, these big rheas that are able to grow to that size, much bigger than most of the birds we see, and to eat the terrestrial foodstuffs, so things like fruits and insects and lizards and things like that. So this was a lifestyle that could have been pursued in all of the different places where the rat-types traveled and eventually wound up. And so that's why they independently could have converged on similar body plans. And this is what has made their evolutionary history so hard to determine. So basically you've got these birds flying to the new locations, and then finding kind of similar types of dinosaur-less, mammal-less areas with similar types of foods. So they all found that the best way to survive in those areas was to grow big and to eat the terrestrial food stuff. And so they all just kind of happened to go that evolutionary direction, and they all ended up happening to look alike. And this explanation also helps explain why the kiwi is so much smaller than the others, because New Zealand is a bit different. Not only is it the smallest of the islands, but it also was already occupied by the moa. So the kiwi had to specialize on other types of food because the moa was already eating that big stuff that the other birds, like the ostriches, would have had a chance to eat. So the kiwi had to specialize in insects, and it also had to specialize into a nocturnal lifestyle in order to have its own little niche that wasn't going to be interacting too much and competing too much with the moa. So now one last bit of science news before I end for the day. And this one is also about something that flies, but sadly not a bird. So I'm going to be talking about fruit flies and how actually they may be a lot smarter than people previously realized and than you likely give them credit for. And this particular study is one that looks at how fruit flies appear to think before they act in a manner that's actually not dissimilar to what primates like humans will also do. And what happened in this particular study is that the researchers put fruit flies into various scenarios where they needed to distinguish between different concentrations of an odor. And sometimes the concentrations were very different, and other times they were very similar. So when they're quite different, obviously that's easy. When they're very similar, that's going to be quite hard. And the experiment involved the chamber was just quite narrow, and it had different concentrations at either end. And at one end was a concentration that the flies had been trained to avoid, and the other was one that they had been trained to know was safe to approach. So as I said, when the concentrations were quite easy to tell apart, when they were quite different, the flies could act very quickly and they could get themselves to the correct side of the chamber. But as the concentrations got closer and the distinctions were harder to make, the flies made more mistakes and they took longer to make their decisions. It was harder for them to tell apart these very similar concentrations. And this longer period of time and this mistake-making process suggested that the flies were basically accumulating information in order to then finally commit to a choice. And this type of behavior has typically been considered something that you see only in animals that have quote-unquote higher intelligence, so things like humans, gorillas, orangutans, etc. And what was even cooler is that when the researchers then developed mathematical models to describe the behavior of the flies, those models were very similar to the ones that had been developed to describe decision-making also in humans and primates. So not only have they kind of seen and observed that the fly behavior is like that of primates, they've also provided mathematical models that support this observation. And all of this decision-making process seems to be related to the activity of a particular gene known as FOXP. And actually there are multiple types of FOXP, but in flies there is only one. And this is found in only about 200 out of 200,000 neurons in the fruit fly brain. So it's quite a specialized thing that only some neurons have. And flies with mutations in this gene took longer to make those difficult distinctions between odors. So it seems to facilitate that hard decision-making process. And that may be because FOXP is somehow related to the evidence accumulation that the flies were going through. So maybe it has to do with memory, maybe it has to do with observing, maybe it has to do with relating something that you've seen to something that you need to do. The researchers aren't yet quite sure. But they do know that in humans who also have FOXP genes, these things are related to lots of different stuff. So language and cognitive development, fine motor skills, so things like piano playing, kind of the range of both physical and mental sorts of things. Now we have 4 different FOXP genes, so it's hard to directly compare, but you get the idea that it is something related to cognition and to basically producing and processing temporal sequences in the brain. So that's how it would have to deal with language, because we know that we have to have certain words in a certain order for them to make sense. Also with piano playing, you have to know, you read the note and then you remember which finger needs to hit that note and in what order to make the actual song. So it kind of makes sense that it would have to do with kind of doing one thing before the other and then lining them all up to ultimately make the right decision in the end. But it also could have to do with circuitry, so just kind of facilitating the interactions between the different neurons in order to allow them to take the decision, take the information from the environment, make a decision, and then make that decision actually come out as a behavior. So there's kind of some fine-tuning that needs to be done with this theory in order to really figure out what role FOXP has in the decision-making process. And of course, I say this all the time, but this does have applications beyond just kind of helping us to understand how things work. So obviously we know that if there are people that have neurological deficiencies where some of these things are difficult, if we can understand how these genes work, then we potentially could develop treatments, even genetic treatments, to figure out how to address that and how to make decision-making and other cognitive processes easier in the future. So this is quite exciting research, beyond just showing us that actually humans and other primates aren't the only smart animals out there. There are lots of species that can do some pretty clever things. Now on that note, I'm going to leave you in the hands of Joan Osborne singing Think, and I will be back to talk to you next week.